overall theme of my lab is structural glycobiology, and that actually involves a lot of areas of research. We study sugars, and we study what sugars do in nature, and we study proteins that interact with sugars and modify them. We're not just interested in the proteins and sugars just as kind of these, ephemeral entities. We're actually interested in them at the atomic level, so the structural level, three-dimensional structures. So we're interested in looking at how exactly proteins interact with and modify sugars by solving the structures of these proteins in complex with sugars, and then we can actually see. It's like getting a snapshot of a car engine and being able to look at it and say, wow, that's how the thing actually works. The natural sciences side is basically where I started out in science. It's an extension of it. And where I started out was studying a class of enzymes called cellulases. And these are enzymes that break down the cellulose in plant cell walls. And they're all the rage because of cellulosic ethanol. And I studied these for many years, and what we ended up transitioning into was kind of a parallel system from the oceans, and that's seaweed. One of the long-term objectives is being able to take that seaweed biomass and convert it into ethanol. So it's the same idea as the terrestrial plants, taking them and turning them into bioethanol, but just using the seaweeds, which is actually, there's some complexities to it, but overall it's an easier problem, we believe, and it's going to be more successful in the end, I think. The reason it's easier is the sugars are in there, the polysaccharides, the polymerized carbohydrates. They're very highly hydrated, so there's lots of water in there. They're easy to get enzymes in, easy to break them apart. And so what we do when we make ethanol from these things is you take the big polysaccharide, you break it down into little pieces, and then you use another microbe, often that's yeast, like you use in beer and wine, to ferment those sugars into ethanol. Now the problem with the terrestrial plant cell walls is the same property of the cellulose that makes the plant stand up is what is the big problem, and that is that they're crystals of cellulose. It's a solid, hard material, and it's really hard to turn that into small sugars, really hard. But we think that the properties of the seaweed polysaccharides, the hydrated properties, are going to make it easier for us to convert them into these fermentable sugars.